to welcome you all here. I mean, I don't know how much of the crowd here constitutes the Bellin Lab, but it's a good crowd. And it sounds cliche, but I think for Burak, I'm going to pull out the bittersweet adjective. And I'll unpackage that in fashion. That's old-fashioned, but I think that there is actually a little bit of a reason behind that. But just reflecting on something I was going to say about Burak, there's tons that I could say, is that it occurred to me that he's actually the first student that I had the opportunity to usher through the whole process. So after I established my lab and got going, I inherited a few students to get going, and they were fantastic, but he was the first one to come in where I had taken a part in his recruitment from interviewing all the way into ushering into a program to joining my lab, seeing him thrive through the developmental biology program, and then seeing him through a thesis. So this is actually kind of on a personal note, demarcates something real significant in my own laboratory also. But for the sweetness and bitterness, there's a little bit of a mix here. So the bitterness, it's a little bit interesting because I'm going to talk about some of the good attributes. It's Burak being the foundation and pillar of my laboratory from the very beginning, with it being the early phases, is that it's actually really shaped the culture of my lab and kind of the overall operational mode, as well as the general culture of the people in the lab. And I think that really boils down to just keep it to a few of some of Burak's remarkable kind of personal attributes. First of all, he's really kind and thoughtful. It's almost to a fault. I mean, he drops anything at any time to help anyone with anything. It's the kind of, that's the way he operates. And, and that goes, it kind of also dovetails right into the fact that he's a natural leader and a natural teacher. So I think probably at least once a week, probably more often, I'd walk through the laboratory and it seemed like Burak was holding court. You'd have at least 3 or 4 people surrounding him, and this was very routine. And it would be people either from our own lab, oftentimes from other labs, either collaborators or completely strangers that I had no idea who they were at all, and just helping them through their experimentation. And that's not to say that he would just do this blindly and without any thought going into it. He's actually bringing to the next phase, he's very rigorous in what he does, and he has a lot of resolve. So in his own experimentation with others, there's another attribute that I'm always fascinated with Burak, is that he digs into these tough experiments. He always naturally knows the proper controls to do, but importantly, and I always stress this to all the graduate students in particular, is that when he starts an experiment, he actually sees it through. So positive or negative on what we started, he answers the question. And that to me is a really, really important thing. Another point that I'll make is that he actually has this very interesting, strong and bold yet subtle independence. So he's not afraid to question authority or to challenge me or Hugo or others around him, but he does it with a lot of respect. And he does it in a way that he doesn't do it to be antagonistic, he does it in a very optimistic way. So this is another feature that I'm always astounded in his operational capacity. And then finally, what I think really kind of encompasses all these little features that I'm talking about and puts it into one term, is that he has an extremely high level of integrity in everything he does. I think all of this leadership, kind of this altruistic operational capacity, and the rigor and honesty, it kind of demarcates integrity. And I think that this integrity is something that I've learned to just completely aspire to this myself, and I think we all should, in the way that Burak approaches a lot of these things. And I think you will see this in some of his talk. Now, why is this bittersweet? This is pretty typical. This is the bitter part, believe it or not. And the reason why I'm a little bit bitter about this is because I think Burak is really the first, and I have never told him this actually, because I never wanted to interfere with his decisions. But he's the first student that has come through my lab where I don't want him to leave. And I mean, I guess we have some say on that right now, but I would actually. So that brings me to the sweet part, and that the fact is that at least for a time being that is ill-defined, we actually don't know how long this may or may not be. He's actually been wooed by Hugo to come into Hugo's lab and work in his lab and carry on his training with genetics, merging some of the mouse stuff that he's doing now with UDN agendas and genetics stemming from flies. And I think he's going to make a huge impact in the whole Bellin Lab initiative by bringing a huge amount of mouse expertise over there. But the sweet part of that for me is that he's still close by. So we can still tap into him, and he still serves as kind of a side foundation over to the edge. So with that, I'm not going to cut into his time any longer, but I just wanted to say to him personally that I sincerely appreciate all of his efforts, not only with his own thesis project and representing the graduate school and the developmental biology program, but I think he's represented my lab really well. So I appreciate it, and please share it with me.
Thank you for the kind introduction. I'll try to meet up the expectations. Okay, so today I'll be talking about part of the work that I have been working on over the years. It became ultimately my thesis work, where we have uncovered the cellular heterogeneity and activity-dependent changes in adult-born neurons in the olfactory bulb. So our brains are every day exposed to many different sensory inputs and calculates this processes this sensory input and controls the complex behavioral output that we are capable of doing. And this is mainly provided by the complicated, complex network of neurons, but the neurons are not working in isolation, and brain ultimately is a really heterogeneous population, composed of heterogeneous populations of cells, including the blood vessel component like capillaries that brings the nutrients and the oxygen to the brain. Also the astrocytes, microglia, and oligodendrocytes, these glia structures that are providing support to neuronal populations. And neurons themselves are not homogeneous either. So when we look at like this drawing by Camilo Golgi years ago, already shows that there's neurons in the brain that have different distinct morphological features, and they are located in different anatomical areas in the brain. And these neurons are serving different functions in the brain, and it is important to understand the function of every single neuron in the circuit to understand how our brains function. And the heterogeneous population of these neurons are mainly generated in the embryonic development of the animal, but for a long time it has been thought that the generation of new neurons ceased to exist in the adult brain. But we now know that there's two regions in the adult brain that has the capability of generating new neurons and integrating into the existing circuit. One of them being dentate gyrus of hippocampus, and the other one is subventricular zone of the lateral ventricles. Neurons generated in the dentate gyrus integrate into the local circuits in the dentate gyrus, but the neurons generated in the subventricular zone migrates towards olfactory bulb and integrate into the olfactory bulb circuit. In our lab, we are focusing on these adult-born neurons that integrate into the olfactory bulb and study their circuit integration and synapse formation. The reason is that olfaction is the primary sensory modality for our model organism. Mouse is using the olfactory sensory cues to find food, evade predators, and regulate their social interactions and even reproductive fitness. So, olfaction is processed in the olfactory bulb in the main, in the central nervous system, and it reaches the olfactory bulb through this pathway where sensory cues travel through the nostrils and inside the nasal cavity and reaches the olfactory epithelium, where each molecule binds to olfactory sensory neurons, and these neurons then project their axons to olfactory bulb and make synapses with the principal neurons of the olfactory bulb, mitral and tufted cells. And these cells receive this information, and after local processing provided by various types of interneurons, this information is sent to the deeper regions in the brain for further processing. And as I mentioned before, olfactory bulb receives adult-born neurons, and some of these interneurons are regenerated throughout the life. The neural stem cells dividing in the lateral ventricles give rise to neuroblasts migrating towards olfactory bulb through rostral migratory stream. And when they reach the olfactory bulb, they became mainly granule cells and populate the granule cell layer. And for a long time, this layer is thought to be composed of single homogeneous population cells called granule cells. But we now know that there are different granule cells that has different anatomical, located at different anatomical layers, and they have different morphological features. And we also know that stem cells located in the subventricular zone give rise to different subsets of these granule cells, adult-born neurons, and different ones, different stem cells give rise to different subsets, and they have unique molecular signatures in the stem cell state. But now the question is whether the mature cells has molecular heterogeneity within them. Based on their morphology and their function, we presume that they have different molecular signatures that identifies them differentially. And we need these molecular signatures to be able to tackle the question, what are the function of each subtype in the olfactory bulb? Apart from being heterogeneous population of cells, adult-born neurons are integrated into the circuit through a tedious mechanism of circuit integration, where adult-born neurons, when they come to olfactory bulb, they go through a selection mechanism where half of the neurons integrate into the circuit and become resident granule cells, and the other half are eliminated through apoptosis. And throughout the studies over the years, people show that this integration process is affected by the olfactory sensory activity, where olfactory increased activity is leading to more circuit integration and survival. So now this leaves us with two main questions that we are trying to answer with our studies. One of them is, what is the molecular heterogeneity within these adult-born neuron populations? And the second one is, what are the molecular mechanisms that translates this sensory activation into synapse formation and survival of the adult-born neurons? So to answer these questions, I'm going to be explaining the studies that I have been working on in two parts. The first part, I will be explaining our unbiased study where we have tackled the cellular heterogeneity and looked at the activity-dependent changes in the olfactory bulb. And in the second part, I will be talking about a developmental signaling mechanism, a novel role for this notch signaling and in regulating adult-born neural circuit integration. So in the first part, to answer this heterogeneity question, we have decided to use a recently developed technique called single-cell RNA sequencing. Doing this, we have identified cellular heterogeneity within the neurons and non-neuronal populations of the olfactory bulb, and we have identified different developmental pathways that these adult-born neurons are using to integrate into the circuit. Moreover, we have also identified that olfactory activity changes these developmental signatures and leads to overall changes in the composition of the olfactory bulb cellular populations. And thanks to Patrick Hunt's graphical artist skills, we have got the cover for the journal that we have published this in. And in the cover, we are showing the cellular heterogeneity by labeling them with different colors. And the whole image is generated by pointillism, which is due to its resemblance to the graphical representation of our data sets. And this, I would like to start by saying that this paper has been a collaboration with James Martin, and Matt Hill was instrumental in these studies. And as you see here, he's wearing a hat, and I tried to find a picture of him without a hat and started going back in his Facebook profile, and I stopped in the first picture. So, I guess he has been wearing a hat since. So, to perform single-cell sequencing, we have taken olfactory bulbs from the adult animals and dissociated these cells into single-cell suspension. And using a commercially available platform provided by 10x Genomics, we have segregated these cells into droplets with a barcoded bead that allows us to identify which cell the transcriptome information is coming from. And these beads also have primers to allow us to synthesize the transcriptome on these microparticles, and also allows us to identify which cell this information is coming from. And after getting these cDNAs, we have amplified it to generate the RNA sequencing library. And using next generation sequencing, we have collected the information about which gene the transcript is coming from and how many of these transcripts are present in each cell. And we have assembled this into a table where we have put the number of transcripts of each gene for each cell that we have captured in our analysis. And using principal component analysis, we have identified similarities and differences between the cells in terms of how many genes are they expressing, what kind of genes they are expressing. And using this information, we have graphically plotted them into these graphs where the cells are similar cells are located next to each other. And when we look at the general populations of the olfactory bulb, we see there's many different cell types. On the left, we are looking at a graph that each cell is represented with a dot, and each color represents cells that are composed of similar transcriptome information. And on the right, you are looking at the genes that are top differentially expressed genes in each cluster, and as you can see that different cell types are expressing different signatures of genes that allowed us to identify what cell type they are coming from. And when we look at the, in general, data, we can clearly identify heterogeneity within many different cell types previously thought to be homogeneous, like microglia and astrocytes. And to our interest, we want to look at the neuronal populations and heterogeneity within them. So that's why we have taken the cellular neuronal cells from this data set, and performing similar analysis, we have clustered them again based on their similarities and plotted them again with our similar graphical representation. And as you can see from this graph, we have 18 different neurons that we have identified in olfactory bulb. So now we want to know what type of neurons these are. First, using classical markers for dividing them into excitatory versus inhibitory neurons, we have looked at which ones are expressed in which cell types. And in this graph, you can see a violin shape in the cells that are expressing this gene. As you can see, SLC17, A7, and A6 are V-GLUT1 and V-GLUT2, which are marking excitatory neurons in the brain, and these are expressed in clusters 15, 16, and 17, telling us these are excitatory neurons in the olfactory bulb, which we know are mitral cells and tufted cells. And as you can already see that we started to see heterogeneity within this population that we have been considering just like two different cell types. And when you look at the SLC32A1, which is a V-gap that is expressed in the inhibitory neurons in the brain, you can see many of the clusters are expressing this inhibitory neuronal marker. And the clusters that are not expressing it, either immature cells or they are different kind of cells like olfactory sensory neurons or the mitral tufted cells. And we have tried to identify molecular signature of every single cluster and identified markers to label these populations. And as you can see, we have identified many genes that are uniquely or in combination defining a certain cell type in olfactory bulb. Now using these markers, we can identify where these cells are located anatomically in the olfactory bulb. Using previously published databases like Allen Brain Atlas Institute hybridization data, we have looked at where each of these markers are expressed in the olfactory bulb. And as you can see already that each marker is labeling a different pattern in the olfactory bulb, which tells us that these cells are located in different anatomical layers in the olfactory bulb. Now we know the molecular signature of these cells and we have identified specific molecular markers for them. We can start studying their function individually by using these genes as a tool to mark and manipulate them. So this brings me to the second part of second question that we are asking using this data set. What are the molecular changes during circuit integration and maturation of the adult born neurons? Adult born neurons, when they reach the olfactory bulb, they go through this stages of morphological changes where they start losing their immature identity, start assembling their synapses, and become synaptically active. And as they transition these different stages, their transcriptome slowly shifts from immature to mature identity. And by using this information, we can look at our data set where we have both mature and immature cells at a given time, because this is a continuous generation of new neurons. We can start ordering the cells into a trajectory where similar cells are going to be close together and the distant cells in development is going to be far away from each other. After drawing this computational timeline, we call it pseudo timeline, and we have realized that this timeline bifurcates into two different trajectories. This tells us already that adult born neurons are utilizing two different molecular pathways that, to guide their circuit integration and maturation. And to know which subtypes of cells are located in which timeline, we have plotted the identity that we have discovered in the previous slides that I have shown. And we have realized that some of the cellular identities are located on one arm of this axis, the others are located in the other arm. So to make it easier for us, we have divided this into two different axes and called it axis A and axis B, and identified genes that are differentially regulated along this maturation process. And to make it easier and save time, I'm going to be only talking about what we have done for axis A, and we have done exactly the same thing for axis B. We have taken the cellular subtypes that are located in axis E and performed the similar pseudo timeline analysis and ordered them again in a trajectory and formed this pseudo timeline for the cells in axis A. And looking at the genes that are differentially regulated along this axis, we have identified many genes that are downregulated and upregulated along the maturation of the adult born neurons. And when we look at the genes that are downregulated and upregulated and look at the gene ontology analysis to see what type of genes are differentially regulated, we can already see that the genes that are downregulated are mostly involved in the neuronal neurogenesis and the cell migration immature identities of the neurons, and the genes that are upregulated are mostly involved in the synaptic transmission and the mature identities of the neurons. So this tells us that our timeline is actually recapitulating the regular, real timeline of neuronal development. And moreover, we have looked at the genes that we already know that are differentially regulated in adult neurogenesis. For example, double cortin is a gene that is regulating the neuronal migration, and it's high in the immature cells and low in the mature cells. You see that in our pseudo timeline, it's also downregulated as the cells mature. And the second example is the corticotropin releasing hormone 1, receptor 1, which is upregulated in our pseudo timeline. And a previous student in our lab identified this gene to be involved in the circuit integration of the adult born neurons, which I'm going to be talking a little bit more about in the second part of my talk. So now we have many genes that are differentially regulated along maturation of the adult born neurons. Now we can start identifying the function for these cells and if they are controlling the circuit integration or survival of the adult born neurons. So in the last question, using the single cell sequencing data, we want to know how does the activity, olfactory activity, affects the circuit integration of the adult born neurons. We know that the increased activity leads to increased survival of the granule cells, but we used to identify them as a homogeneous population. Now we have subtypes, whether each subtype is affected by the olfactory activity similarly or they are differentially responding to olfactory activity. To manipulate olfaction, we have used two different methods. One of them is being loss of function. The other one is going to be our gain of function manipulation. For loss of function, we have closed one nostril of the mouse to block the sensory input from reaching only one side of the olfactory bulb. And using that, previously people shown that the occluded side is getting smaller because of the increased cell death and decreased integration. And by labeling these adult born neurons and following their fate for 4 weeks of olfactory deprivation, people previously shown already that this method is reducing the survival of the adult born neurons. So this is going to be our loss of function in our experiments. And as a gain of function, we have utilized our previously published method where we have trained the animals to discriminate two different odorants. They are both exposed to this odorant and they have to pay attention to identity of the odorant, which makes them engaged to receive olfactory stimulation all the time. In this case, mouse in a behavior box needs to poke their nose to the center port to receive an olfactory cue, and based on the identity of the cue, they have to go to the side port to get a water reward. And if it's a go odor, they go get their reward, and if it's a no-go odor, they have to wait for the next trial. And previously, this has been also shown to be increasing the survival of the adult born neurons in the animals that are engaged in discrimination tasks. So using these two methods to manipulate the olfactory activity, we have performed this olfactory manipulation for 6 weeks in a different set of animals and collected the olfactory bulbs and dissociate them into the single cells and perform single cell sequencing to compare the cell numbers and for the survival changes in the adult born neurons. After isolating the adult born neurons and labeling them based on which sample they are coming from, and as you can already see that the cells coming from different manipulations are located in different layers, different parts of this graphical representation. And when we look at the cellular identity and plotted that into our in our data set, we can identify that some cellular subtypes are increased in the trained sample, and some of them are increased in the occlusion samples. So you want to quantify this, and it's by using the assumption of if there's no change in the cellular number, it will be equally composed of wild type, occlusion, and the trained samples. And using this information, we have performed chi-square analysis and calculated the chi-square values and used that as a value to calculate increase or decrease in the cellular numbers. And when you look at the occlusion samples, you can see that some cellular identities are increased in the occluded animals, and some of them are decreased in those animals. So by converting this into a binary system, by saying that increased the cells that are increased are going to be labeled red, and the cells that are decreased are labeled blue, we plotted this into our pseudo timeline, and to our surprise, we have realized that the cells that are located on one axis are behaving completely opposite to the cells located on the other axis of development. So this is indicating that the cells that are using similar developmental pathways are behaving similarly when upon olfactory manipulation. And we analyzed the same, analyzed this data for the training samples as well, and we have realized that we have seen a completely opposite changes in the cell numbers. So the cells that are increased in the occlusion are decreased in the training. The cells that are decreased in occlusion are increased in the training. So this reciprocal olfactory activity changes are changing the cell numbers in the adult-born neurons reciprocally. And these cells are located in different axes in the developmental pathway, which argues that these pathways are affected by sensory activation oppositely. So in the first part of my talk, I have shown you that we have discovered that the cells in the olfactory bulb exist in different, demonstrate different heterogeneity within themselves, like microglia and astrocytes. We already identified different heterogeneous populations of cells for these populations. And we have identified specific molecular markers to describe adult-born neurons in the olfactory bulb. Now we can start tackle the question of what each neuronal subtype is doing in the olfactory bulb. We have also identified that adult-born differentially, and the cells are changes in the cellular survival is changing the cellular composition in the olfactory bulb. So in the first part that I have shown you right now, I have been talking about the cellular heterogeneity and adult-born neuron survival. In the second part, I will be talking about a novel mechanism for a developmentally studied signaling mechanism, notch signaling. As I have shown you before, adult-born neuron survival is activity dependent, and half of the neurons integrate into the circuit, but the other half is eliminated through apoptosis. And we know that by looking at the genes that are differentially regulated along this maturation process, we wanted to identify mechanisms for circuit integration and survival. And one of the genes that we have identified from our data set was corticotropin-releasing hormone 1, which we have studied previously to be expressed in the adult-born neurons along maturation and integration. And Isabella also showed that the knockout of this receptor is leading to reduction in the survival of the adult-born neurons in the olfactory bulb. So this shows that this gene is upregulated in as the cells mature and regulating the circuit integration and survival. And she showed clearly that this molecule, corticotropin-releasing hormone, is released by the local interneurons in the olfactory bulb and acts on the adult-born neurons, which expresses the corticotropin-releasing hormone receptor 1 and leads to increased synapse formation and circuit integration. So now we want to know what ties the activation of this receptor into synaptic integration into the circuit. To answer this question, we have performed a microarray analysis where we have taken samples, olfactory bulb samples from corticotropin-releasing hormone knockout animals and compared it to wild type and the animals that are expressing a constitutively active version of this receptor. And when we compare the gene expression profiles from olfactory bulb tissue, we have realized that the many components of notch signaling pathways are differentially regulated along between these manipulations. And we know that the notch signaling is regulating neuronal maturation at different stages of development. So it has been shown to be involved in cell fate decisions, differentiation of the neurons, and regulating the maintenance of the stems, mediator of circuit integration and survival. So notch signaling is initiated by ligands that are expressed on the signal sending cell. And these ligands are membrane-bound molecules from two different families, jagged and delta-like one. And they bind to their receptor that are expressed on the signal receiving cell. And mammals are expressing 4 receptors, notch 1, 2, 3, and 4. And upon interaction of the ligand to receptor, this receptor is cleaved by gamma secretase and releases the intracellular portion to travel towards the nuclei and binds to RBPJ and turns it into an activator and initiates the transcription mechanism to express downstream target genes, which are mostly Hess and Hay genes. And the first question you ask whether the notch signaling is even active in the olfactory bulb. Using a transgenic animal where 4 RBPJ binding site driving the expression of a nuclear localized fluorescent molecule, we asked only in the, this molecule is going to be expressed only in the cells that the notch signaling is active. And we looked at the brains from these animals and realized that the olfactory bulb has a high level of reported expression. And when we look closer, we have realized that the granule cell layer, which contains mature adult-born neurons, have really high level of notch reporter expression. And when we look at the rostral migratory stream, where the immature cells are located, the level of notch signaling is really low compared to the mature cells. To make the distinction between the mature and immature cells more clearly, we have labeled the mature cells with the neuronal nuclei, nu-N, and immature cells with double cortin DCX. And as you can see from here, that the immature cells located in the RMS almost have really low level of notch activation, but the mature cells have increased levels of notch signaling. And we know that by going from rostral migratory stream towards superficial layers of olfactory bulb, granule cell layer, we know that the granule cell maturity is increased. So there's more mature cells in the superficial. We know that the granule cell maturity is increased. So there's more mature cells in the superficial layers and less mature cells in the deeper layers. And when we look at the notch reporter activity along this axis, we can see that the notch reporter expression is also increased as we go to superficial layers. So this tells us that the notch activation is correlated with the maturity of the adult-born neurons. So, but we want to know at what point adult-born neurons initiate the notch signaling. Is it correlated with the circuit integration? To answer this question, we have performed a lineage tracing experiment where we have injected notch reporter animals with a DNA intercalating agent, EDU, and labeled the adult-born neurons when they are born and follow their fate at different time points, whether they have a notch signaling active or not. And when we look at the data, we have realized that the adult-born neurons, as they mature, more of the adult-born neurons initiate notch signaling. And it plateaus after the circuit integration process is finished. So this tells us that the adult-born neurons initiate the notch signaling during this circuit integration time frame. But is the notch signaling even activity dependent, or does it depend on olfaction? To answer this question, we have taken the same notch reporter animals and performed similar olfactory deprivation assay where we have blocked one of the nostrils to block the activation only on one side of the bulb, and the other side is spared to receive a sensory input, and looked at the notch reporter expression level in closed versus open side of the olfactory bulb. And as you can see from the pictures right away, that there is a clear difference between the open side versus closed side. We have quantified this using intensity measures on the granule cell layer. We have seen, we can see here that it's decreased in the olfactory deprived side of these animals. So this is showing us that the notch signaling is also olfaction dependent. So increased notch activity is correlated with increased olfaction. So we wanted to do the next experiment to assay the loss of function of notch signaling by removing, using two different strategies. One is we removed RBPJ to block the transcriptional activation of the downstream targets. So, but we know that the notch receptors can also interact with the molecules in the cytosol, and this leads to different functions of the Notch receptor called non-canonical pathway. To block also the non-canonical pathway, we decided to remove the receptors, and this blocks the non-canonical, and also because of the loss of intracellular portion, this also blocks the non-canonical pathway. So, but which one are we going to be knocking out? Because there's 4 different receptors expressed in the mammalian genome, and we started looking into the databases to see which ones are expressed in the olfactory bulb. And we have performed antibody staining for Notch1 and Notch2. We have realized that Notch1 is expressed ubiquitously in the forebrain of the animals, and Notch2 is restricted to immature migrating adult-born neurons. And the Notch3 has been shown to be not expressed in the brain, and Notch4 is mostly localized to the vasculature of the brain. So now we know Notch1 and 2 are the targets that we want to knock out. We have acquired conditional alleles for these animals, where these receptors, or in the RBPJ case, RBPJ gene is flanked by LOXP sites. Upon expression of the Cre recombinase, this gene is removed from the genome and it cannot be expressed anymore. So we have decided to deliver the Cre expression using viral approach into the migrating neurons, Russell migrator stream, and using mixture of viruses. One of them is expressing Cre recombinase together with a GFP molecule, and the other one is expressing a TD tomato TD tomato fluorescent marker only. And after 4 weeks of circuit integration, we have quantified the number of cells that survive at the end of the integration, and get the ratio for between Cre and non-Cre expressing cells in all 3 transgenic animals: wild type, RBPJ conditional allele, and Notch1 2 double conditional animals. When we look at the ratio, we can already see that the RBPJ flux animals have increased number of cells when we knock out RBPJ gene. And when we look at the Notch1 and 2 flux animals, we see even more increase in the survival of the cells. So this indicates that the Notch signaling acting through both canonical and non-canonical pathways to regulate the survival of the adult-born affected. But how does these neurons the circuit? Are they functionally integrated? Are they receiving are they receiving synaptic input in the circuit? To answer this question, we have focused on our attention on the Notch1 and 2 flux animals because that one gave us more robust phenotype, and we wanted to focus on both canonical and non-canonical pathway removed case. And using similar strategies to knock out these receptors in adult-born neurons, we have injected viruses and waited for weeks to knock the receptor out, and performed in vivo in slice electrophysiology to record number of electrical synaptic activities these cells are receiving. And here you can see 2 example traces. One of them is coming from a TD tomato positive control cell, and the other one is coming from Cre expressing GFP positive cell that the Notch receptors are knocked out. And we quantify this data by looking at the number of synaptic activities and the frequency of this activity, and also the amplitude of each synaptic input, and compare them to each other. And you can see from this graph that the conditional knockout cells, where the Notch1 and 2 is knocked out, the cells are receiving less synaptic input in numbers, but the synaptic input levels in terms of amplitude is not changed. So this is telling us the cells that Notch1 and 2 is knocked out, surviving better, but they receive less synaptic input in the circuit. To look at how does this affect the principal neurons, mitral and tufted cells, we wanted to knock out Notch1 and 2 in all the inhibitor neurons in the olfactory bulb. Using this Cre driver, DLX56 Cre, which is expressed in the forebrain inhibitor interneurons, we have decided to knock out Notch1 and 2 receptors using this Cre driver. And as you can see in the olfactory bulb, adult-born neurons, when they reach the olfactory bulb, they are still expressing double cortin, and they initiate the Cre and then knock out. Normally, in this case, it's expressing TD tomato, and they are going to be knocking out Notch receptors only at the end of their circuit integration. And then we performed electrophysiology on mitral and tufted cells to see how these cells are affected from knocking out receptor in adult-born neurons. here you are looking at two example traces. One of them is coming from our control animals, notch 1 and 2 flux animals, and the other one is coming from notch 1 and 2 knocked out animals using DLX563. Again, looking at the frequency of these synapses that mitral and tufted cells are receiving, we have quantified the frequency and amplitude of these synapses, and we have seen an increase in number of synapses that the mitral and tufted cells are receiving, but the amplitude did not change. So this tells us that the adult-born neurons are providing more inhibition on the mitral and tufted cells, probably due to increase in the survival, increase in their survival and integration to the circuit, which we should test in the future. So in the second part, I have shown you that the notch signaling is active in the adult-born neurons, and it is also olfactory activity dependent, increased activity leading to more notch signaling. And when we knock out notch signaling using two different strategies, we have seen that its activity normally regulating the adult-born neuron survival negatively. And when we look at the synaptic integration, we see that the notch mutants have less synaptic input. And when we knock out notch in the adult-born neurons, we see that the mitral and tufted cells are receiving more inhibitory input, probably due to increased survival. So I have shown you two different parts that I have been studying, and these two parts gave us the answer of the molecular heterogeneity within the neurons of the olfactory bulb, and we identified a novel pathway, a novel mechanism for a developmental, classically defined pathway. Now, based on this, we know that the morphologically distinct neurons also expresses different molecular markers. Now we can match these morphologically distinct neurons with our molecular roadmap and start building tools to mark and manipulate these cell types and study their unique contribution to olfactory bulb circuit. Moreover, we have a bunch of genes that we have shown differentially regulated in adult-born neuron maturation and integration, and now we can study the function of these genes, whether they are correlated with the circuit integration or survival of the adult-born neurons. So only then we can start understanding how this complex network of neurons in our brain can carry out these complex functions in our daily life. So thank you, and go ahead. So I just want to say a couple of thanks to a couple of people that helped me throughout the studies. I already thanked my collaborator, Jim Martin and Matt. I would like to thank my mentors that guided me along the way and bared with me with changes in my projects that I have been going through. That I have changed a lot of projects, and this one became my thesis work. And I learned a lot, and I learned a lot from them. And the good thing is they have distinct approach to science. I have seen two different ways of doing science in one PhD life, which is an advantage for me. I would like to thank both of you guys. I would like to thank all my lab mates that not just scientifically, but also we have spent time every day in our lab. I've probably seen them more than my wife. It's over 6 years. And eventually we became a good team that overcome many different challenges. One of them was against the NEAM lab. We overcame successfully those challenges as well. So I'd like to thank my committee members. Again, they know that how many different projects that I have been going through, and how many changes happened in my PhD, and their endless support and really appreciated input helped me reach to a final project that became a really nice story. And I would like to thank all of you guys for their input and support. And I would like to thank again Hugo and Catherine, because they recruited me to DB, and this is the only place that I got accepted. So But if it wasn't the case, I would probably choose here. And but I'm sure that Hugo is affected by my previous PI, Arzu, who was my first mentor, and I'd like to thank her for her and the support and letting me go out and explore. And many of the other people in our department was asking for people to stay and have their PhD in Turkey, but Arzu was supporting us to go out, explore the science in other parts, and I would like to thank her as well. I would like to thank all my friends that I acquired along my PhD, including DB friends, drinking friends, camping friends, friends from ATA Houston. So, of course, without them, this would be like, I don't know. I can't even imagine having no friends in like a distant place apart from your family. And I would like to thank them for making this journey a pleasant journey for me. I would like to thank my family again for their endless support. My sister here was the first one who suggested me to pursue a career in science, and she made me start this like journey. That's why I would like to thank her to support that, because she was supporting me to come here to United States as well. And I would like to thank my parents, and I'm gonna say this, that because of this choice that I have made in my career, every time that I changed the school, it made me go further and further away from them. Even though this was the case, they supported me without any question, and I would like to thank them for their endless support. And last but not least, I would like to thank my best friend, my wife. And as you can see from these pictures, we have been together for a long time. This is our first picture actually, and it's from like 11 years ago. And I would like to thank her for her endless support and following me all the way here. And I remember she once told me that she would never leave Turkey, and yet she came here, followed me all the way here. And I'd like to thank her because she's been adding more value to my life every day. And I'm sure in the future she's going to be adding even more value. And the next edition is going to be coming in September. And we are going to be having a baby boy. And I would like to thank her for, again, endless support and giving me the world. And do you have any questions?
Wonderful talk. I think it's a great. Both projects are great. I'm sitting here. I need to ask a notch question.
Yeah.
The non-canonical requirement of notch, I think it's interesting. Is that ligand dependent? Do you know? Is it?
None of the studies that we have performed is ligand specific. So I checked a couple of like expression patterns of the ligands. It seems like JAG1 and delta like1s are expressed in adult borneos, but the JAG2 is expressed in the mitral cells. So right now we don't even know which cell is the signal sending cell, and is it ligand dependent? We need to first answer like which ligands is at play, and then start going towards that problem.
And maybe one more related question. The axis A and axis B, does notch signaling change across those two axes?
That's a good question. I wanted to like look at many of the notch pathway components in our dataset, but because of the nature of the signal cell sequencing, we have really shallow sequencing information, and the notch signaling components are really expressed in low levels. So we don't see any of the receptors or ligands expression in our dataset.
Thank you.
Thank you.
No other question? Oh, there you go.
Just a pretty basic one. The last data points that you presented, it kind of looked to me like there was a little bit of heterogeneity in the sense that there were like sort of two groups which were kind of clustered, and looked like the significance was being driven by maybe 2 or 3 outliers. And it looks like that maybe suggested some heterogeneity.
So you mean the single cell sequencing? Yeah. So
the very last data points that you were showing.
Yeah, I think like this is not the whole heterogeneity. This is like definitely broadly defining the cell type. So you mean this one?
No, keep the very, very last one, the very, very last thing, the plots, the data plots. Just keep going right to the end.
And
Physiology.
you mean the notch stuff? Or
Yeah, right at the very end.
Okay.
Yeah, sorry. So these ones, so you kind of see these like two tightly clustered groups, and you have these couple of outliers which seem to be driving the significance. So just kind of wondering if you think that's a real effect, or if there's something different about those outliers, or.
So like in wild type, like in the frequency.
Yeah, on the left.
If you remove those 2 points.
Yeah.
red
Yeah.
points,
Yeah,
they're
so
the same, right?
yeah, like right now we don't have statistical power to say these are outliers. So by adding more samples to it, we can see whether they are actually outliers or like are they part of like the gradient of variation. So I can't say anything right now, but like when we performed outlier tests, they were not like excluded, probably because of the sampling rate.
Any other questions? Okay, with that, we'll be having a happy hour in about an hour on the 10th floor, I think, of the NRI. So why don't we all thank Burak again for a fantastic job.